Once again, I explain culture of protoplast. Here, in culture of protoplast, we want to use MS liquid medium. That MS liquid medium is nothing but Murasic and Skoog medium used with the some modification in droplet plating or micro drop array technique. I already explained plating method for the culture of protoplast. Then the protoplast viability that can be able to tested with the help of fluorescent diacetate before the culture, because the fluorescent diacetate that can be able to give the viability nature of the protoplast. After the protoplast that can be able to incubated in continuous light, especially 1000 to 2000 lux at 25 degrees Celsius. Then after the incubation of the protoplast, the cell wall again it can be able to form. That is we want to say regeneration of cell wall. That regeneration of cell wall occur nearly 24 to 48 hours. That is 1 to 2 days. And first division of new cells also occur between within a week. That is from 2 to 7 days of the culture. Then next one is selection of somatic hybrid cells. What is the meaning of selection of somatic hybrid cells? With the help of a protoplast, the result is nothing but somatic hybrid cells. That somatic hybrid cells that can be able to develop a entire plantlet. That entire plantlet that can be able to develop into a plant. That is called it as a somatic hybrid cells. Fusion product of protoplast without nucleus of different cell, that is called it as hybrid. Hybrid means fusion of protoplast with cell wall, that is called it as hybrid. Here fusion of protoplast without nucleus of different cell, that is called it as hybrid. Following this nuclear fusion happen, that entire process is called it as somatic hybridization. That entire process is called it as somatic hybridization. With the help of isolation of protoplast, fusion of protoplast, culture of protoplast, we want to obtain a somatic hybrid plants in our biotechnology. Next one is called it as, based on that culture, the next culture is called it as cell suspension culture. The cell suspension culture is nothing but CSC. C means cell, S means suspension, C means culture. That what is the meaning of cell suspension culture? Growing of cells, including the culture of single cells or small aggregate of cells in vitro in liquid media. Simply I said growing of cells. That growing of cells, we want to select a single cell or a group of cell. Small aggregate means group of cells, especially in laboratory condition. We want to use the MS medium in a liquid medium without agar medium. That is called it as a cell suspension culture. Here we want to prepare a cell suspension culture with the help of a rotatory shaker instrument. Here there is a rotatory shaker instrument. Here there is a conical flask. That conical flask, they contain a MS liquid medium. That MS liquid medium, we want to inoculate the single cell or a small aggregate. That single cell or a small aggregate that can be able to agitated or shaking with one another. That shaking or agitated, that the cell suspension culture that can be able to develop or form a callus in a liquid media, that is called it as a cell suspension culture. Here mostly we want to use the cell suspension culture for the production of secondary metabolites. Here production of secondary metabolites in our day-to-day life that can be able to used for our daily activity of the human, especially the cells are separated from the callus tissue and used for cell suspension culture. This diagram is called it as a laminar airflow diagram. This is called it as a centrifuge. Here the laminar airflow chamber, we want to use the in vitro conditions for the development of the liquid media, callus, embryogenesis, etc., or isolation of protoplast, fusion of protoplast, all the one they can be done under the laminar airflow chamber. The laminar airflow chamber is one of the instrument that can be able to uh, done the protoplast culture or any other culture in aseptic condition. That biotechnology work that can be able to occur under the laminar airflow chamber. This is called it as a centrifuge. That a centrifuge that can be able to used for to separate the protoplast as well as debris. The debris is nothing but the unwanted waste. That is also a one type of instrument. Then next one is called it as production of secondary metabolites. CSE, cell suspension culture, can be useful for the production of secondary metabolites like alkaloid, flavonoids, terpenoids, phenolic compounds and recombinant proteins. These are the some of the secondary metabolites that can be able to developed with the help of the cell suspension culture. Here, what is secondary metabolites? Secondary metabolites are chemical compound that can be able to segregate in the plants naturally, but they are not required by the plant growth, but that plant growth and development. But the secondary metabolites are the byproduct of the plants for the cell metabolic activities. Secondary metabolites are not used for the plants for the normal growth and development, but it is the byproduct for the cell metabolism. For example, biosynthesis and isolation of indole alkaloids from Cathanthus roseus plant cell culture. For example, Cathanthus roseus is nothing but Nithya Kalyani, Tamil we say Nithya Kalyani or Sudugatu Malli. Here, the root of the Cathanthus roseus, they can have the capacity to contain one type of alkaloid, indole alkaloid. The name of the indole alkaloid is nothing but vincrustine, that can have the capacity for the anti-cancerous property. Then, the process of production of secondary metabolites can be scaled up and automatic using bioreactor for the commercial production. Mostly here secondary metabolites are used for the commercial production. Then next one is many strategies such as bioinformatics, elicitation and immobilization have been used to make cell suspension culture. That cell suspension culture are more efficient in the production of secondary metabolites. Here some of the few examples that can be able to given in the table below. For example, secondary metabolites like digoxin, codium, capsin, vincrustine and quinine. Here what are the plant source and what are the uses for the secondary metabolites? Digoxin, that can be able to obtained from the plant Digitalis purpurea. Mainly it can be used for cardiac tonic. Then next one is call it as codeine. Codeine is otherwise call it as morphine, that can be able to obtained from Papaver somniferum. That Papaver somniferum is nothing but casasa, that can be able to used for analgesics, mayakam tharakudiyadu. Then next one is call it as capsaicin. That capsaicin that can be able to obtained from the capsicum annum, melaha, that can be mainly used for rheumatic pain treatment, mootu. Then vincrustine, I already explained. Cathanthus roseus, anti-cancer carcinogenic agent. Then last one is called it as quinine. That quinine that can be able to obtained from the Cinchona officinalis, that can be mainly used for anti-malarial drug. Not only for that alkaloid that can be mainly present. Most of the alkaloid that can be able to present in our day-to-day life activity, that can be mainly used for the in our human life activity.